Welcome back to Enzymes Day 2. So today's aim is what increases enzyme activity. And after today's lesson, you should be able to name either 2 or 3 things that increase enzyme activity, or 2 or 3 factors, I should say, that increase that activity. So before we get started, I want to ask you 2 questions to jog your memory about yesterday's lesson. The first one is, what is an enzyme? And the second is, what is a substrate? So go ahead and flip back to your notes from yesterday if you need them, and take 2 minutes to answer those 2 questions. Pause this video as you answer them and press play when you're ready to go over the answers. So question number 1, what is an enzyme? You should have written something like this. Enzymes are proteins that speed up the rate of reactions that would otherwise happen more slowly. We also call them organic catalysts. So we call them organic because, once again, they are made up of carbon and hydrogen because they're proteins. And the word catalyst just is a fancy way of saying it speeds something up. So you may have heard the word catalyze before, which means to speed something up. That's the same thing we're talking about when we say the word catalysts. So catalysts catalyze reactions. They speed up those reactions that would otherwise happen more slowly. And remember, again, since they are proteins, they are made up of amino acids. All right. Now, what is a substrate? Substrates are the specific chemical that each specific enzyme acts upon. So one important thing to remember is that enzymes have one specific purpose. So enzymes have a specific purpose or function. All right, and that purpose or function is going to be influenced based on their substrate. What are they supposed to be acting on? What's that specific chemical that they are supposed to bind with? Now, I know that the bottom part is covered up, so I'm going to press done real quick so you can copy that down. The specific chemical that each specific enzyme acts upon. And then if you want to add my notes that I've written, feel free to do that as well. All right, let's keep going. So we're going to skip these questions. Now, we've got some notes that we need to fill in. So first off, yesterday I mentioned something called the active site or the binding site, but I didn't dive into too much detail. So today we need to learn a little bit more about that. So something you need to write down is that enzymes are usually much bigger than their substrates. So we'll see a picture of this coming up shortly, but in most pictures, you'll see enzymes. Oh, that was an ugly shape. You'll see enzymes represented something like this and substrates represented like this. So as you can see, the enzyme is a lot bigger than the substrate that it connects to. So the protein chains fold to make a 3-dimensional shape, or 3D, that has a specific pocket or site where the substrate can fit and interact. So if we're looking at this picture right here, our active site is going to be right here. So this is going to be that site where the substrate can bond. And once again, the specificity of the enzyme is dependent on its active site, specifically the shape of its active site. So depending on what shape that side of the active site is, influences which substrates are able to bind. Let's say for a second that this is I'm going to use the same enzyme here. So we've got a little triangular active site. I can't draw shapes, but the site or the shape of this active site is going to influence what can or cannot bind to it. So if a little square substrate came along, it's not going to be able to fit on this active site. That's not going to work. I mean, maybe if it turned on its side, no, it's not going to work. Same thing if we had let's go crazy and say that we've got let's just do a circle. If we had a circular substrate try to come in, it's not going to fit because that active site does not align to the substrate. So the active site is going to influence what substrate is able to bind to the enzyme. All right, let's keep it rolling. So here I've got a picture for you as an example. So here we can see this is going to be your enzyme. Okay, this big old thing. And this is your substrate. So once again, you can see that the enzyme is a lot bigger than the substrate is. The substrate is tiny when compared to that giant enzyme. But this pocket right here is going to be our active site or our binding site. And you can see that it's the same shape as our substrate. So that circular substrate is going to be able to fit right there inside of our active site. And then what happens is this chemical reaction takes place, right? That enzyme actually closes around the substrate and allows that chemical reaction to occur. So it closes off, and then that's where the magic happens. All right. So, this is dictated by what we call the lock and key theory. I did touch on this yesterday, but I'm gonna go in a little bit more detail. So as I said yesterday, each enzyme is specific for one and only one substrate. So we call it the lock and key theory because it works pretty similarly to how locks and keys work, right? You have to have a specific shaped key to enter in your door. If it's not the right shape, that door is not gonna open. If I try to take my key and go open up your house door, it's not gonna open, right? So the key has to match up with the lock in order for it to open or work. And that same thing happens with enzymes. So enzymes and substrates have to have the same shape in order for that chemical reaction to occur. So I like to think of it as like puzzle pieces. Let's see if I can draw a puzzle piece. Oh, no, go back. Ah. All right, I like to think of it as like puzzle pieces. So, I already know this is not gonna happen. All right, so this is our enzyme. Our substrate has to connect perfectly. That is a horrible picture. I'm so sorry, guys. So I should not do art. That's what I've just learned from that picture. But I like to think of lock and key theory as like puzzle pieces, because the edges of those puzzles have to line up perfectly, just as the enzyme and the substrate do. So those shapes have to be the exact same so they can match up together and get the job done. If those shapes don't match up, they're not going to work. Okay, so we've got one lock and one key. Now, it is thought that in order for an enzyme to affect the rate of a reaction, the following events must take place. So on your paper, you have got 4 steps of the enzyme substrate complex theory. You don't have to write anything down, but I am going to walk you through each of these steps. I would highlight before we go on that each enzyme is specific for one and only one substrate. Let's go ahead and highlight that on your paper. All right, moving on. So enzyme substrate complex. So whenever we have an enzyme matching up with a substrate, it's going to look something like this. You've seen different renditions of this picture, but we've got an enzyme which is much larger than the substrate. And then you've got this little pocket where the substrate is going to fit into. And remember, they have to have the same shape. And we're going to call this pocket the active site or the binding site. Okay, and once that substrate matches up with the enzyme, it's going to fit into that little pocket and form what we call the enzyme substrate complex. And I'm here to show you how that happens. So the first step of the enzyme substrate complex is the enzyme forms a temporary association with the substrate or substrates. So you can see that in this picture over here. This substrate is going to just come down and fit into this tiny little active site on the enzyme. It's going to nuzzle itself into there and form a temporary association, which just means they're going to momentarily connect. Okay, that's the first step. Second step is the enzyme and substrate form a close physical association between the molecules called an enzyme substrate complex. So this is where we actually see those two particles connecting and forming that enzyme substrate complex. And you can see in this picture right here, it actually says that the enzyme changes shape slightly as the substrate binds. So whenever the substrate connects with the enzyme, that enzyme is actually going to close in around that substrate to hold it in place. So it gives it a little hug. So hold substrate in place like a hug, which we can't do anymore, rip. All right, so the enzyme is then going to hold that substrate in place, forming the enzyme substrate complex. Third step, while the enzyme substrate complex is formed, the enzyme action takes place. This is where the chemical reaction happens with that enzyme. The substrate is going to be broken down into its smaller parts. So for example, a disaccharide is going to be broken in half and become 2 monosaccharides. So the enzyme actually breaks that substrate in half. Okay, so you can see here that originally where we had the substrate represented as green and as a whole picture, in this second part right here, we actually see it changing colors. Now, the substrate is not actually going to change colors, but this is to represent that it has been broken apart and is now 2 separate entities. So the enzyme breaks that substrate in half. All right. So I'm gonna go ahead and write, so like if this is our disaccharide, then our enzyme is going to break it apart into 2 monosaccharides. Okay, so the third step is the enzyme breaks that substrate in half. And then the final step is after completion, the enzyme and the product separate. The enzyme is then ready to react with another substrate. So you can see that these substrates, after they've been broken apart into their monosaccharide forms, are just piecing out. They're just fleeing the scene. The damage has been done, except it's good damage. And the enzyme is going to go react with another substrate. So as soon as this has happened, the enzyme is going to just be recycled and it's going to start this process all over again. So this process repeats. And remember, it's able to repeat because the enzymes are never used up. So enzymes are not used up. They're actually recycled. So the enzymes are used over and over and over again to do the same thing. So they break a substrate, move on, break a substrate, move on. They're almost like little heartbreakers, right? Bouncing from one substrate to the next, okay? They're just there to break everyone's heart in half. All right. Now, a few more notes for us to take. Oh, but here's one more picture showing that. So step 1 is the substrate. In this example, we've got sucrose. Binds with the enzyme on that active site. They form that enzyme substrate complex. The enzyme is going to break the substrate apart into glucose and fructose. Then the products are released and the enzyme goes back and does this over and over again. All right, if you have any questions about this, please let me know. All right, now enzymes, remember, as I just told you, are not changed during the reactions and can be reused. So we note that only the substrate is broken. The enzyme is not broken. I'm actually gonna write that down. So only the substrate is broken. Not enzyme. So the enzyme is able to be recycled and reused, but that substrate is broken in half. All right, moving on. So I'm about to show you guys some factors that influence the rate of enzyme action. The first one, I'm not sure why this is so small, is pH. So the first factor you need to write down in your notes is pH influences enzyme action. So the best the best pH for most living things is close to 7, which is a neutral pH. So if we go much higher or lower than 7, it usually slows down the enzyme activity and has them work slower. And especially, well, I'll get into that later, but there are a few exceptions to this. There are a few enzymes like gastric peptidase that actually work best at acidic pHs. So work best at a pH of around 2. So if we were to represent this as a graph with enzyme activity and pH, we would see that action looks something like this, where as pH increases up to 7, we see enzyme activity increasing. But the second if we pass 7, that enzyme activity is going to decrease again. Okay, so this would be 0, this would be 14. So the optimum activity, the best activity, happens at a pH of 7. All right, the next factor that influences the rate of enzyme activity is temperature. So temperature strongly influences enzyme activity. The best temperature for enzyme activity is around 35 to 40 degrees Celsius. And it's similar to pH in that it's going to proceed slowly below optimal temperatures. And actually above 45 degrees Celsius, that enzyme is actually going to get destroyed. We can also call that denatured. So the enzyme essentially falls apart. So once again, if we were to represent this as a graph, it would look similar to that pH graph where we see enzyme activity on the left axis and temperature down here. And let's say we've got 0 degrees Celsius, 100 degrees Celsius. So as temperature increases to around 35 or 40, we're going to see enzyme activity increasing. But then after we pass 35 to 40, it's going to start decreasing again. So the optimum temperature, or the best temperature for enzyme activity, is going to be around 35 to 40 degrees Celsius. Okay, so I'm going to put best, fastest activity. All right. And the third factor that influences the rate of enzyme activity is actually not on your paper. So you don't need to write this down, but it is something you need to be aware of. And that is the concentration of enzymes and substrates. So when there is a fixed amount of enzyme and an excess of substrate molecules, the rate of reaction is going to increase to a certain point and then level off. So this leveling off occurs because all the enzyme is used up and the extra substrate has nothing to combine with. So I'll show you guys what that looks like on a graph. Essentially, it would look something like this. So once again, enzyme activity. With concentration, or I'll say like amount of substrate. So it would look something like this, where the activity would increase, but then once we run out of substrates for that enzyme to connect with, the activity is going to level off. And it's not going to increase, it's not going to decrease, it's just going to stay the same. And that's because there is no more substrates for the enzymes to connect with. So it's kind of like dancing, okay? Once everyone has found a partner and has partnered off, if there's anyone left standing, there's no one else for them to connect with. So the same thing happens with enzymes and substrates, where once everyone has found a partner, once all those enzymes have connected with a substrate, the activity is going to level off. It can't increase, it can't decrease. And then the same thing, if more enzyme is available than substrate, the same type of reaction will occur, where it will increase and then level off. So the excess enzyme will eventually run out of substrate molecules to react with. All right, and that is everything you need to know today. So right now on your worksheet, you need to follow that, follow the link to the exit ticket, fill out that exit ticket as always, and press submit. That is how I know that you did today's lesson. If you have any questions, let me know. Send me a message on Remind and I will get back to you shortly. Have a wonderful day and I'll see you guys